Hi, my name is Dr. James Woods. I'm a senior research scientist at Jordi Labs. Today we're going to cover some of the analytical services that Jordi Labs can provide for you. So the agenda for today is, who is Jordi Labs? What makes Jordi Labs one of the premier analytical service companies in the world? And what can we do for you and your customers? We're going to do that by covering a few example analyses. Jordi Labs was founded in 1980 as a GPC analysis lab. This is a gel permeation chromatography. It's used for determining the molecular weight of a polymer. And since then, we've really blossomed into a full-service polymer analysis lab. We perform extractables and leachables analysis. We do identification and quantification for medical devices, for food packaging, for pharmaceutical companies. 80% of our employees are degreed chemists. We perform a lot of investigative analysis, polymer and identification, as well as deformulation and discoloration studies. A few of the problems that we solve are related to polymer analysis. We can do extractables and leachables, good-bad comparisons. Particle characterization is really something that we're great at. Method development and validation for pharmaceuticals, for example. Polymer filler and additives, as well as product deformulation, quantitative analysis, and contract research. And one of the things that we really pride ourselves on is the ability to do most projects within 10 business days. So if time is of the essence, we're able to deliver results on time. So some of the ways that we use to accomplish this are our core analytical services. That includes mass spectrometry, chromatography, what we're founded on, molecular weight analysis by GPC, spectroscopy, including FTR, SEM, NMR, as well as elemental and thermal analyses. So from that, one of the tools that we use is QTOF instrumentation. We have both QTOF GCMS and QTOF LCMS instruments. And QTOF stands for quadrupole time of flight. And this is a technique that's used to provide ultra-high mass accuracy. The analyte fragments go through a time of flight instrumentation where very precisely the mass of the fragment is able to be recorded. And that's important because it allows us to use very powerful algorithms to identify the molecular formula with scores to how confident we are for that match. And we're then able to use library searches for reference spectra, including NIST and Jordi proprietary reference spectra, to identify what these components are. And if that doesn't work, then we also have fragmentation spectra, which we're able to fall back on and effectively piece together what the original component was. And so, to sum it up, we have extensive GC QTOF and LC QTOF expertise, and we're able to identify more unknowns than most other places. When you know what it is, but you want to know how much of it is, that's why you would use a triple quad mass spec. That's QQQ if you're in that parlance. So a triple quadrupole mass spec provides ultra-high quantitative accuracy, and this is accomplished by taking the analyte, mass selecting for that particular analyte, fragmenting it on the second quadrupole, and then using the third quadrupole to be mass selective for the fragments of that particular analyte. And this is an excellent technique for definitively quantifying a component. And we have multiple ionization sources. It's very good for small molecules, small organic molecules. And we have extensive experience with triple quadrupole mass spectrometry. Another technique, because we're a full-service polymer analysis lab, is PYMS. PYMS is pyrolysis gas chromatography mass spectrometry. It's a technique that effectively depolymerizes your polymer sample using a very small sample amount, and provides not only the structural identification of the polymer itself, but also of the additives that are present in the sample. So this is a technique, if you're looking at the slides right now, where, for example, we can compare a good and a bad resin pellet. And we need a very small amount for this, maybe 1 milligram of sample. And we're able to identify, for example, in the good pellet, the presence of ergophos 168, and in the bad pellet, the presence of oxidized ergophos 168 and the absence of unoxidized ergophos 168. And so it's a very powerful technique for polymer analysis. Here's another slide for it. For identifying the additives that are present in a polymer sample, as well as each pyrolysis, each pyrogram provides detailed information about the base structure of the polymer. So you'll see differences in isotactic versus syndiotactic polypropylene, for example. It's a very powerful tool for polymer analysis, and we're really privileged to have it and offer it to our customers. Like I said, we started as a GPC analysis lab, and so molecular weight determination by GPC is still one of our core strengths. We provide not only high temperature GPC, which is great for polyolefins like high density and low density polyethylene, as well as polypropylene, but also we have instruments that are dedicated to specialty solvents. We have more than 30 GPCs, and of that we have several that are dedicated to HFIP, we have several that are dedicated to DMSO, and so if you have a polymer system like nylon, which goes into HFIP, we have instruments that are dedicated to that. We don't have to switch an instrument over. We have expertise with aqueous systems. We have a ton of applications for GPC, cellulose, for example. So here's a few example analyses of, in this case, I think low density polyethylene in TCB. Using our brand new Resolve column line, we developed our own column line, our Resolve column line, which is monodisperse. In this case, it's a 13 micron particle size for the stationary phase inside, and we have some pretty good refractive index and right angle light scattering for low density polyethylene. Here we have high density polyethylene using very similar conditions, and you can see in this case it's at 160 degrees Celsius, so this certainly classifies as high temperature GPC. And we have, like I mentioned, several instruments which are dedicated to not only high temperature GPC, also specialty solvents. And in addition to that, we also have tetra detection GPC. This is a really big advancement where it uses multiple detectors to identify the molecular weight of a sample without a standard. So suppose you have a research grade polymer, which there is no appropriate standard for that for standardized GPC. We can determine its actual size in solution and figure out the molecular weight from that. So it's a very powerful technique. Other techniques that we have include spectroscopy, and this could be FTIR microscopy or standard FTIR, as well as NMR for structural elucidation. We also perform UV-vis spectroscopy. And some of the applications for this include FTIR microscopy mapping. So if you have like an engineered surface or a multi-layered film, we're able to perform polymer analysis, functional group analysis, and mapping on that structured surface. And multi-layer films are one of our specialties, where we're able to take the multi-layer film. I think we just did one recently for like a 7-layer film. It was pretty neat. And we were able to take it on edge and identify the chemistry of each of the layers on edge. Another thing that we really pride ourselves on is particle analysis, and I'll get into that in more detail later through a few example analyses. But I guess the takeaway for this is we can image features all the way down to 15 microns using FTIR microscopy. And complementing that, we also have SEM EDAX, which can go down to surface mapping sizes of 5 nanometers or so. It's a very powerful tool, not only for the surface morphology, but also for the EDAX, the EDX analysis, where we're able to do elemental analysis on that as well. And we have specialized tools that assist us for this with SEM EDAX, because we're a polymer analysis lab. We have a gold sputter coater, for example, to dissipate any sort of charge that builds up while we're doing that. And so we can do an unknown identification and particle analysis with our FTR, with our SEM EDAX, and as well as surface mapping. Another tool in our arsenal is ICP-MS, and that stands for inductively coupled plasma mass spectroscopy. It's a great technique for elemental analysis. So if you want to know what elements are present in whatever your sample is, this is one of the go-to tools for that. And our particular ICP-MS is pretty unique. It can, in one analysis, it can provide not only trace but also bulk elemental analysis. So if you have something that's sitting there at, say, 20%, like a titanium oxide, we can also we can identify the titanium oxide with confidence at 20%, and we can also identify whether or not lead or arsenic or other trace elements are present down in the part per million, part per billion range. It's a robust instrument that is able to tolerate up to 25% total dissolved solids. And we have specialized techniques, again, because we're a polymer analysis lab, to quickly and completely digest samples so that they're ready for ICP-MS. Because it's one thing to have an ICP-MS that's capable of doing this, but on the other hand, the wet chemistry that goes into preparing a sample for ICP-MS is very important, because otherwise you could get ion suppression and other techniques. So complementing that, we have different kinds of thermal analysis, including TGA and DSE. And so we can provide the melt point, the glass transition temperature, the percent crystallinity, as well as using our modulated DSA, we're able to determine the reversing versus non-reversing thermal transitions in a sample. We can also perform TGA, it's thermogravimetric analysis, and that's a technique where you heat up a sample and you look at the mass change as a function of the temperature that's coming off of that. And so using that, we can determine the residual volatile content, as well as identifying degradation temperatures and filler levels, that sort of information. And we have this really cool hybrid technique called EGA-TGA, where we take a mass spec and we run it through the same. We run the sample through the same temperature profile as our TGA, and you end up, because you're running the same temperature profile both in the TGA and your EGA, you get a mass change from your TGA to tell you how much, and you get different peaks that are eluting off of your EGA, your evolved gas analysis. And so that tells you how much and what it is, which is something that most labs are not able to do. Again, one of the things that we pride ourselves on is particle characterization, and some of the sources that you could get for particles where it's really important to identify what the particle is. Packaging for pharmaceuticals, potentially undissolved residuals in like a bioprocessing or pharmaceutical manufacturing. System components could be present in there. You could find gaskets or seals. You could also find side reaction products, for example, charred products, detergent residues. And then also you could find corrosion or lubricants, other issues with scoring on your product line. And it's very important to identify the source of these particles. For example, if you have a regulatory agency that's interested in ensuring good quality patient care, for example, depending on your drug product or something like that, not having contamination in it. And so some of the tools that we use to identify these particles, for example, if it's the bulk organic composition versus the bulk inorganic composition, we might use FTIR microscopy or SEM-EDAX. These are both non-destructive techniques and probably where we would start in most cases, because we don't want to damage the sample, and it's often a very sample-limited case. You might only have like 2 or 3 particles tops. We've had cases where it's just 1. And then moving on from that, we have we can provide more information about the detailed organic composition using techniques like POMS, that's pyrolysis mass spectrometry, or QTOF LCMS. QTOF GCMS is another option if we're interested in more volatile species. But unfortunately, these are all destructive techniques, and so if we're sample limited, we'd probably want to start with a non-destructive technique. If you're interested in the detailed inorganic chemistry, for example, if it's like a rust or corrosion problem, we could start with maybe ICP-MS, which unfortunately is also destructive. But it is very sensitive, down to the part per billion, in some cases lower. And so here's a case study where we go over some example data for particle characterization. And in this particular case, I believe it was from a bioreactor. Yes, it was from a steel bioreactor. And they gave us a residue and a brown particle. And so the samples in this case are descriptively labeled as brown particle and brown residue. And so here's an optical micrograph of the brown particle and the brown residue with a 2 millimeter optic bar on it. And then here's a zoom in on the optical micrograph of the brown particle. And so it's got, at a closer scale, it looks like it's a white material with some brown residue on it. We don't want to jump to conclusions, but I think we'll pretty conclusively show what it is in a little bit later. So in this particular slide, on the left side, we have the optical micrograph that we've done at higher resolution, and we've mapped together all of the imaging. And on the right, we have an overlay of iron oxide in green, and then also of PTFE signals, that's fluorinated ethylene absorbance in red. And so we're starting to draw some conclusions from this. It looks like it's some PTFE with some iron oxide on it. And then what we did to follow up with that is some SEM-EDX. SEM-EDX is a great technique for elemental composition, and you can determine, you can do surface mapping with SEM-EDX. And what we've done in this case is we've confirmed the presence of PTFE and iron oxide using SEM-EDX. And so the EDX map that you're looking at right now, the overlay of fluorine is in yellow, and then iron, that's Fe, is kind of a reddish orange. And so there's a ton of fluorine that's present, which would be expected for PTFE. And there's traces of iron as a reddish orange. And here is the EDX-derived spectra, as well as the elemental concentration of a few spots on the brown residue. And so we can see a really nice spectra that's consistent with iron oxide and a little bit of carbon. We see some K-lines for carbon. And it basically confirms our previous analysis that this is a PTFE particle with some iron oxide on it, and that the brown residue is actually just iron oxide. And so following that up, here's an FTIR overlay of the brown residue in blue, and then iron oxide standard. So we have standards of most kinds of material, not only in our database, but also reference standards of them available to us to image. And it matches quite nicely. So we're pretty sure that this is iron oxide. And in this case here, we have our brown particle. Our brown particle overlay with a PTFE standard, and we also have, I believe, a separate area on the particle. And so they overlay quite nicely, and we can see some good signals to confirm this for us. And then to really drive it home, to show off how cool the instrument is, we have an FTIR heat map. And so this is a technique where we image different spots on the sample, and we're able to put those together with software and construct a heat map. And so you can see on the left there's the iron oxide spectra, or sorry, the spectra that are consistent with iron oxide are on the left, and they map as red for more of this there, while on the right we have the PTFE signal. And it's pretty conclusive that it's a PTFE particle that's been coated in rust through the bioprocess. So it was, the customer liked this data, and it pretty well answered the question what this was. Moving on to another kind of example analysis that we do, polymer discoloration. In this particular case, a syringe manufacturer came to us, and they had one lot of syringes that was yellowing before they should have. And so on the top you see a clear, unadulterated syringe. Looks good enough to use. The one on the bottom is yellow, and it should not yellow. So they wanted us to figure out what was the cause of the yellowing agent. And so in this particular case, we ended up dissolving the syringe. So we took the whole syringe, we dissolved the entire polymer, and then we precipitated out the polymer itself using a co-solvent. You can do this in most cases with like a chloroform hexane or some sort of solvent system. It's a trial and error sort of process. But what you're looking at now in this case is in the pan for each case is the yellowed and unyellowed control sample. And so we dissolved the syringe, like I mentioned, and then precipitated out the polymer. And so the polymer is present in the precipitated polymer is present in the aluminum pan. The liquid which we used to precipitate out is present in the scintillation vial, and then we took that and concentrated it down for both cases so that we have a control and our yellowed sample. And that's present in the HPLC vial to the next to that. And because we're looking for a yellowed sample, it's very important that our HPLC vial actually contains a colored sample. Otherwise it might be something like a pigment, which we would expect to be not soluble in a solvent, and so because it was colored, we decided to move forward with LC-MS analysis. If it was uncolored, we might start by doing elemental analysis on the polymer sample neat, because a pigment wouldn't normally dissolve in an organic solvent. And so after injecting this on our LC-MS, one of our LC-MSs, I guess, this one was equipped with a UV vis, and I believe it also has a CAD detector as well. We see an unknown colored species that comes out quite early in our colored sample, but we don't see it in our control. And I think in the next slide we're able to identify several EIC peaks that are present, that are coming out at that same time. What we're then going to do is we're going to look at the isotopic distribution of these samples, or sorry, of the eluting peak. From that, because we have a QTOF instrument attached to this particular LC system, we're able to identify with high mass accuracy what the isotope, I guess the mass of each fragment coming out is. And then we're able to pass that through. An algorithm that determines the molecular formula for that particular mass. And in this particular case, we're able to identify the component that's present on the screen right there, and we're able to provide that information to the client, and they're able to look at what they were doing to formulate this particular polymer system, and they were able to identify two different components that were cross-linking in their polymer system over time when exposed to UV light. And they were able to eliminate that from their polymer system and move forward with production for this syringe. So another example analysis that I'd like to get into is polymer cracking. So this particular client came to us and they wanted to know why their polymer system was cracking. And so a couple of different reasons why that could potentially happen. When they're sourcing the material, it could come with other components that are a problem. So the raw material sourcing might be a problem. It might be a problem associated with the production process, or it might be use in the field, like when you're actively using the product. And so this right here is a GPC chromatogram where we took a sample of the virgin resin, a sample of the processed resin, as well as two of the used products. They were both used in different use cases. And we were able to determine the molecular weight of the polymer in each case. And that's important because cracking is typically caused by decreased molecular weight. The long polymer chains end up reinforcing the final part, and you're able to have a more resistant material. And so what we're able to show in this case here is that the molecular weight of the virgin resin, of the processed resin, is effectively unaltered. So the raw material is fine. The production process is fine. Where we start seeing a problem is after the product's been used in a few different ways. And so the black trace and the orange trace, in this case right here, show a decrease in molecular weight of the product. And so this informs our next decision of, do we need to identify the additives that are present in this sample? What's the next thing that we have to do to fix this problem? And so a couple of follow-up analysis for this particular case might be pyrolysis mass spec or LC-MS to identify the additives package that are present in this. And that's ultimately what this client decided to go for. And we were able to identify the additives package and then show that we needed to improve the additives for this particular product. So those were a couple of the smaller pieces of how Jordi Labs can help on small projects. But we excel both at large projects and at small projects. And so some of those larger products are typically regulatory in nature. So analysis in support of regulatory filings, method validation for those regulatories, and then also extractables and leachables testing. For example, well, I'll get into that. So what are extractables and leachables? You may not know what they are, but if you don't, so they're chemical entities that come out of a pharmaceutical package or some sort of component that's regulated under laboratory conditions. And so these are conditions that could accelerate or exaggerate the normal conditions of storage. In contrast, leachables are also chemical entities, but they're extracted under the normal conditions of storage or use. And they're typically, but not always, a subset of extractables And so typically what you'll do is perform the extractables analysis first, and then use that to inform your leachables analysis, because one is a subset of the other. And so who are the kinds of companies that need extractables and leachables analysis? This is going to be biomedical device companies, this is going to be food packaging companies, pharmaceutical packaging, anything where it's a regulated environment and it could impact patient safety. That's patient or consumer safety, I think, is an appropriate umbrella for that. And so some of the guidance that we have for that includes stuff like ISO 10993. There's a whole host of ISO regulations for that, rather ISO guidance for that. The FDA also has some guidance as far as, for example, 510K medical devices. There's also some European guidance for that, as well as different USP standards for pharmaceutical packaging. So as I mentioned before, leachables are typically, but not always, a subset of extractables. And some of the components that could be in extractables and leachables include anything that was present in the polymer system from the beginning. And that could include antioxidants, slip agents, surfactants, plasticizers. And so anything that you put into a polymer system that potentially gets implanted into a patient or is used for a patient could end up getting bioaccumulating or migrating into the patient. And it's important to be able to use techniques which are appropriate for polymer analysis if you expect to analyze things which are related to polymer analysis. And so how we do extractables and leachables is an important component to that. It starts with an expert regulatory project manager. We have project managers who specialize in FDA guidance. We have project managers who specialize in ISO guidance and REACH guidance. They're going to thoughtfully consider the sample, how it's used. They're going to discuss with you what are the best ways to do the extraction? What are the best ways to do concentration? How do we do quantitation? And then they're going to work with you to design a robust and complete study. And so after we have that study, we're going to clearly communicate the benefits and trade-offs before we do the analysis. And so it's important to have consideration for how a study is going to affect the results in the end, because you may decide on one particular extraction condition, but there are trade-offs for that, and hopefully we'll go over that in a second. So they'll also oversee the preparation and the analysis itself, and then on top of that we're going to clearly communicate the results back to you. We're not just going to data dump and give you a bunch of numbers. It's going to be a complete report, and we're going to have a sit down and we're going to discuss the results that we obtained for you. Here's a couple of here's an overview of our extractables and leachables thought process. We put a lot of thought into how we design an extractables and leachables analysis. And so at the beginning of an extractables and leachables study, we consider things like the device composition, how the device is used, and then also when we're doing study design, what are the most appropriate analytical techniques for this sample? How we're going to select an appropriate sample, and then how we're going to extract this sample. And so before we even do a study, you have to put in this work to get appropriate results. And then while you're in the process of performing the analysis, there are 4 key steps to this, and that's the extraction, the concentration, identification of the components that you extract, as well as the quantification of those components. And we put a lot of thought not only into the study design, but also into those 4 key steps, because how you do the extraction is important. It affects the results. How you do the concentration step. Are you going to miss any volatiles when you do your concentration? How you do the identification, that's very important, because the fewer unknowns that you have in your final study, the more likely you are to pass your tox screen. And then how you do your quantification is also very important as well. And so to kind of illustrate that point, we have, I guess in this case, a milk carton. How are you going to do the extraction for a milk carton? Do you want to take the whole milk carton and submerge it into your test fluid? Probably not, because then you're going to extract out the inks that are on the outside of the package. Most appropriate case in the most appropriate extraction conditions for this particular use case is probably to fill it up with your simulated use fluid and extract just the polyethylene that's present in there. In other cases, you may want to extract just one side that's exposed to a patient, for example, like a dermal patch or something like that. We've actually developed techniques to do a single-sided extraction, which didn't exist before we had that particular project. And so I guess the takeaway for this is we put a lot of thought into how we do the extraction, how the sample is going to be used, to generate the best possible data, the most appropriate data for that extractables and leachables study. So that way, when you submit this data to your regulatory body, it's the most complete data, it's justifiably the best data that you can get, and it's good quality science. And so the extraction conditions that we use for this are informed by how the device is going to be used. How you extract a device is you can get very wildly different results depending on how you perform an extraction, and depending on the temperature, on whatever solvent strength that you're using, if you're doing several different kinds of solvents, how long you extract something, those are going to affect the extractables that you get out of your final study. And then your leachables are usually going to be the most stringent use conditions that you get. Sorry. The next step for that that has, I guess, the most important concern is how your samples are concentrated. So if you have volatile analytes and you take your sample to dryness and then you perform an extractables and leachables study on that evaporated residue, you've missed anything that's volatile. Anything that has a boiling point above, I don't know, say 50 or 60 degrees Celsius, it's gone to atmosphere when you blew it off. If you wanted to really identify everything that's present in the patient when the device is exposed to the patient, how you concentrate your sample, it matters. And then also how you perform controls and validation. Like when we perform an extractables and leachables analysis, we include a blank that's gone through all of the same processes as the sample, but it just doesn't have the sample in it. And we're able to compare that to the actual sample extraction. And it's essential to a good quality study. So the ENL analysis flow starts with the screening analysis, where we get the gist of what's present in the sample. That could be a gravimetric or a total organic carbon, determine the pH, and then it follows up with a qualitative analysis where we identify what's present in the sample. There's a whole host of techniques that we would use for that. And then after we have our qualitative analysis, we follow that up with a quantitative analysis using chromatography and mass spectrometry. And in this case, it's really a quantitative analysis where we identify exactly how much of whatever analyte is present. And how we decide on an appropriate instrument in this case depends in large part on the instrument selectivity. So different instruments have different instrumental limits of detection. And so, for example, headspace GCMS, dynamic headspace in particular, is a very powerful tool for volatile components. You can get 1 part per billion limits of detection for headspace GCMS, but it's only good for volatile components. On the other hand, LCMS or GCMS may be a more appropriate technique depending on the volatility of the analyte that you're looking for. And so, deciding on an appropriate instrument to perform an extractables and leachables analysis, you don't want to decide on just one. You want to use a broad spectrum of instruments to more appropriately cover the entire range of expected analytes that you're looking for. And so screening analysis is very important to use, again, like I said, this broad selection of instruments. But then once you've identified what's present, to then hone in on the appropriate technique using the appropriate, for each appropriate analyte using the appropriate technique. And so what this slide here illustrates is, for an example, extractables and leachables analysis. We may have all of these different analytes that are present here, and deciding on the appropriate technique boils down to the physical differences between the analytes. And so, for example, dimethoxyethane and hexadecane are great for headspace GCMS, but on the other hand, we have to use something like QTOF LCMS for ergonox 1010. Or maybe even uruclamide. And so you can identify and quantitate different analytes using different techniques, I guess, is the takeaway from this. So, like I mentioned before, one of our competitive advantages is the fact that we use QTOF instrumentation not only for LCMS but also for GCMS. We have several of each. And so what that allows us to do is to use commercial databases like NIST and Wiley's databases, as well as our own proprietary additive and oligomer database, which we've developed over the history of the company, to identify unknowns effectively and quickly. So the QTOF instrumentation provides this high mass accuracy. We're able to use an advanced algorithm to do molecular formula generation for each unknown that comes out. And we can also do mass spec fragmentation using our QTOF LCMS or chemical ionization using our QTOF GCMS. And what used to be an unknown is now a known. And I don't know if you've done a tox screen before, but when you do a tox analysis for most US-based regulatory bodies, what they require you to do, if you have an unknown, is to lump them all together in the same toxicological evaluation. And so you may have 35 unknowns, you do a mass aggregate of each of those unknowns, and that one set of unknowns may cause your tox analysis to be over limit. What happens at Jordi Labs is we're able to identify significantly more species. There are fewer unknowns. You're able to do a targeted toxicological analysis for each particular analyte, and you have a significantly better chance of passing your tox analysis. So here's an example where we perform QTOF LCMS identification, foreign extractables and leachables. I think this is compound 21. There's probably 20 or 30 other compounds that were extracted out in this particular ENL. And looking at the high mass accuracy that we get out of a QTOF instrument, we're able to pretty definitively show that this is steramid. You can see that 284 peak for steramid right there. And it has a really good match score for this, and we're pretty confident that this is steramid. In the case that we don't have a good mass match for this, we can also look at the mass spec fragmentation pattern using our QTOF instrumentation. And in this particular case, we're looking at the mass fragment for the compound that's shown on the screen. That's N-lauryl diethanolamine. And what we see here are several different fragment ions of the parent structure, as well as the best match formula for each of those. And we're able to, I guess, put the pieces back together and figure out that this is N-lauryl diethanolamine. And so getting back to more the 10,000-foot view, an extractables and leachables analysis also includes quantitative method development. Once you know it's steramid, for example, you have to figure out how much is present. And so you've identified the components, now you have to quantify them. And so we have some of the best chromatographists in the world that work at Jordi Labs, and we do method validation daily. And so we can develop methods for most unknowns. And so, So quantitative method development is a component that goes into extractables and leachables analysis. Once we've identified what a component is, we have to quantify it. And so there's a few different ways that we can use to do that. We can develop a validated analytical method if it's something that has a high concern. Or alternatively, we can use other techniques, for example, formal quantitation or relative quantitation, using an authentic reference standard or a surrogate standard, if that's appropriate. And I'll go into a couple of examples of when it's appropriate to use a formal quantitation standard or a relative quantitation standard. So on this slide right here, it's a little busy, but I'll break it down for you. So the different analytes that we're looking at right now are on the x-axis, and for example, ethylbenzene. And what we've done is we've quantified each of those analytes using a reference standard, using the same reference standard. And those reference standards are below. They're all deuterated versions. For example, deuterated toluene, deuterated chrysene. We have phenanthrene and naphthalene. I think they're deuterated analogs. And so this kind of highlights both the strength and the weakness of relative quantitation. It's very useful if you need a number for a tox analysis. And most tox analysis They apply a safety factor on top of whatever value you get out of your extractables and leachable study, and so it's okay. But if you have a component that is fairly toxic, or you need to know exactly how much is present, then you can see the benefits of formal quantitation. And so, for example, taking the ethylbenzene example, if we use toluene as a reference standard for ethylbenzene, we're going to significantly overestimate the amount of ethylbenzene that's present. On the other hand, if we use a formal standard of ethylbenzene, it's going to be right on the money when we're doing our formal quantitation. And so it's kind of this trade-off between if we have 85 components that we need to quantify, we can use relative quantitation in those cases, but if there's 2 or 3 that are very important, it's important to determine exactly how much is present, then maybe we want to use formal quantitation for that. And so we have several different kinds of quantitative strategies depending on the needs of your study. Some of the limitations mean that you need a high purity analytical standard for formal quantitation. And so in some cases that's just not possible. If it's, for example, an oligomer or some other specialty chemical or something that's formed in the degradation, you may not be able to get an analytical standard for that. And so other cases that you could use for that might be an external standard or standard addition. So I guess in summary, a successful study has 4 key points, and that's the appropriate extraction conditions, careful concentration of the extracts that are present in the sample, definitive identification of the components that we find, and then also accurate quantitation. And so those are the 4 key steps that we value at Jordi, and hopefully for your samples to come. Here's a brief list of the techniques that we offer at Jordi Labs. It's pretty comprehensive, and again, we seek to serve you with modern chemical analysis and solve your problems. We want to make it our goal to not give up until we solve your problem and provide the best data set possible for your project every time. Thank you very much, and I'll see you again throughout the day.